Okay, so welcome to this webinar series on microglia. I'm Guy Brown from the University of Cambridge and my co-host is Claire Butler. So this is a series consisting of monthly talks of about 45 minutes from European scientists talking about their research on microglia, followed by a Q&A session. So in relation to the Q&A session, if you have a question that occurs to you during the talk, please just put it into the chat function and we'll read them out in order at the end. And can I ask you to keep your microphones off and your video off during the talk. Okay, so I'm gonna hand over now to Claire, who's going to introduce our speaker.
Yes, hello everyone. So our speaker is Sally Cowley from the University of Oxford. So a brief introduction on Sally's past work. So after Sally graduated with a degree in natural sciences here at Cambridge University, she began her research career working on host pathogen interactions in mycobacteria at the University of London. For postdoctoral work, Sally focused more on signal transduction pathways involved in cellular differentiation. After taking a career break to raise her children, Sally joined the Sir William Dunn School of Pathology at the University of Oxford as a Wellcome Trust Re-Entry Fellow in 2007, working on differentiation of human pluripotent stem cell-derived macrophages and their applications in HIV studies. Later, with William James, she set up the James Martin Stem Cell Facility at Oxford, which specialises in work with human pluripotent stem cells and houses great collaborative projects, such as developing iPSC microglia to study microglia's contribution to neurodegenerative diseases, and also developing a large-scale cellular drug screen for iPSCs derived from around 500 Parkinson's patients. However, here today, she's talking to us about using human pluripotent stem cell derived microglia to model neuroinflammation. So take it away, Sally.
Thank you very much. Thank you so much for inviting me to speak in this seminar series. I think it's a fantastic initiative and, you know, particularly in COVID times, but in ordinary times as well, for us to all get together and enthuse about our favourite cell, microglia. So before I get deeply into the talk, if you just want a 3-minute sort of cartoon version of the kind of stuff that we do in our group, then follow the link at the top of this page, which will take you to a video made by Hazel Hall Roberts in our group, who's a very talented postdoc whose work I'll be talking about later in the talk, who won a competition to make this outreach video. Okay, here we go. So there are microglia are the resident macrophages in the brain, and they carry out many homeostatic functions, including clearing up of dying neurons and trimming away of incompetent synapses and mopping up of misfolded proteins. This is their normal homeostatic function, but if they become inflamed in any way, because they are a professional immune cell, they will spit out inflammatory cytokines, and if this is not resolved in a timely fashion, then it can be damaging to the surrounding neurons. So it's the sort of balance between these functions that need to be very finely tuned and is part of what goes wrong during neuroinflammation and neurodegenerative diseases. So there are many different genes of neurodegenerative diseases that are involved in macrophage and microglial functions. For Alzheimer's disease, about 40% of the genes associated with the disease are associated with microglial and macrophage functions. So this includes several phagocytic receptors such as TREM2 and its downstream signaling components, and complement receptor 1, CR1, and CD33. And they can also take up the misfolded proteins that are characteristic of this disease, so amyloid beta and MAP-tau. In motor neurone disease, or ALS, C9orf72 is a disease-causing gene, and this is expressed in macrophages and microglia, and it's been shown to be a RAB GEF, so it's involved in the shuttling of vesicles within the cell. And then in Parkinson's disease, alpha-synuclein is the misfolded protein that is characteristic of Parkinson's, and this can be taken up and processed by microglia. And all of these misfolded proteins can potentially be spat out in partly degraded forms if the microglia is not behaving itself. Also, within the cell, we have the lysosomal protein GBA and also LRRK2. Which are Parkinson's associated genes, and they're expressed in microglia as well. So today I'm going to talk to you a bit about how we have developed models of macrophages and microglia that we can apply to investigating what the functions of some of these gene products are. And so in the first part of the talk, I'll describe the models, and then I will talk in the second part of the talk about work we've done on TREM2. And then in the final part of the talk, I will show you some work that we've been doing on LRRK2. So, why bother with IPS macrophages and microglia? Well, there are many reasons. So, existing cellular models include transformed cell lines such as THP1 and BV2. However, they're karyotypically abnormal. You can see from this karyogram of THP1 cells that it has several trisomies, and this is clearly not an appropriate model for modeling diseases that are where gene dosage is really, really important. So for example, in Parkinson's disease, a triplication of the alpha-synuclein locus can lead to early onset Parkinson's disease. And so that's just a doubling of the amount of alpha-synuclein that's present in the cells. Further, these cancerous cell lines, they don't terminally differentiate properly, and their metabolism is that of a cancer cell, so that's not appropriate for modelling the metabolism and differentiation properties of cells like microglia that are effectively terminally differentiated. The mouse has been a fantastic model, especially for looking at microglial function in situ. But microglia from mice lose their identity as soon as they're taken out of the brain and put into culture. And very importantly, not all of the pathways are conserved or regulated in the same way as the human. And so, for example, 15 out of 44 of Alzheimer's genes have no ortholog in the mouse. And an example of this is complement receptor 1. The ApoE polymorphism that is characteristic in man is completely absent from mouse, and TREM2, the R47H mutation, which is the most common TREM2 mutation in Alzheimer's in man, that has a splicing defect in the mouse that is different than in man, and so it means that the mouse is not really appropriate model for that. Human primary microglia are extremely hard to source, either fetal or adult microglia, so that's not usually a viable option. And then using blood monocyte-derived macrophages is also not a very appropriate way to go, because they have a completely different ontogeny to microglia and they can't be genetically manipulated very easily, as I'll show you in a minute. So for all of those reasons, we decided over a decade ago that we would like to develop a methodology for making macrophages, initially and subsequently microglia, from human iPS cells, so that we could understand human macrophage biology better, and in a system that would be authentic and genetically manipulatable. So we can genetically manipulate iPS cells using CRISPR-Cas technology, and we developed a very straightforward way of differentiating the iPS cells to macrophages in a sort of two-step process. This has been the sort of workhorse of our lab ever since, and has been adopted by quite a few labs around the world as well. So the attraction of this method is that it produces a lot of macrophages, and by that I mean we've been able to scale up to over 1 billion to be able to do genome-wide CRISPR screens and drug screenings. We know that these cells are going through a primitive hematopoietic differentiation program, because if we knock out c-Myb, which is required for adult definitive hematopoiesis, then it has no effect on the ability of these differentiation cultures to produce macrophages. They still produce as many. So this is a Myb-independent differentiation process that's going on in the dish. And this means it's a great starting point for modelling microglia, because microglia are, they start off life in the yolk sac. As primitive macrophages emerge from the yolk sac in early embryogenesis and migrate into the developing embryo to populate all of the tissues of the body, to become tissue-resident macrophages, including the brain. And within the context of the brain, they mature and become the sort of adult microglia that you're used to looking at. And so this is the sort of correct ontogenic pathway that we should be trying to recapitulate. So we can take our differentiated macrophages, we can either plate them as macrophages, or we can skew them to being more microglia-like by giving them neuronal environment-specific cues, either by in monoculture or more convincingly in co-culture with iPS neurons. And this can this co-culture can become more complex as well. We can add in astrocytes as well to make it more authentic still. And this was the work done by Walter Hanseller in our lab. And this was published a couple of years ago. We were not the only ones to publish at that time. There was a complete flood of papers all around the same few months describing methods for making iPS microglia. The basic early differentiation part, mimicking this kind of primitive hematopoietic or myelopoietic pathway, is pretty common to all of them, although I would argue that ours is probably the more efficient and productive of them, and simple. The cocktails that people use for their final plating of the cells is different, and also whether people are specifically focusing on monoculture or co-culture in 2D or in 3D organoids. Depending on the kind of questions that they're asking, these different models are more or less appropriate. And very excitingly, most recently, it's been possible to put these cells into, engraft them into mouse brains, and they adopt even more convincing microglia-like properties when that's done. So what of our cells that we are producing, our iPS microglia, when we compared them with fetal microglia, primary microglia, then they cluster very nicely with them, and they cluster away from blood monocytes and away from the macrophage early precursors that we have, partway through the differentiation process. When we look at the most common set of human microglia markers as defined by Butowski and also by Bennett, we can see that if on each of these graphs we have blood monocytes on the left-hand side and we have fetal microglia on the right-hand side, and then are progressively more skewed towards being microglia cells across the middle, and we can see that as the cells are skewed more and more to being microglia-like, then they increase in their expression of all of these core set of microglial markers. But it's important to note that even the macrophages, our standard macrophage condition, that already has high expression levels of most of these genes. Because it's already a primitive macrophage that's kind of on its way to becoming more defined as a tissue macrophage. So what about their behaviour? So this IPS microglia are in red, and they're sitting on a bed of IPS cortical neurons. And here we can see that they roughly tile across the field of view. And they seem to have adopted territories, and this is very reminiscent of what we see in the brain. And within these territories, they're surveying the territories really actively and interacting with the neurons in a very proactive way. And if we look at the rate of change of their ramifications, this video has one frame every 12 seconds, and you can see that the ramifications are changing with every frame. So they're incredibly dynamically surveying their neuronal environment. We can also observe in these co-cultures phagocytic events going on. So if you look at the green arrow, you'll be able to see that this green microglia is really interested in this red cluster of neuronal cell bodies, and it keeps revisiting it, and after several visits to that region, it phagocytoses something. So that is good evidence of phagocytosis in a dish, though it's very hard to quantify. And so we often resort to being in monoculture to be able to observe these phagocytic events. Here's a macrophage that is about to phagocytose an alpha-synuclein particle that's tagged with 488. In it goes. And if we run the video again, we can also see this cell is about to die. And it's immediately consumed by its neighbours. And so this is a really typical homeostatic behaviour that goes on in tissues, almost unobserved under normal conditions, because you're normally just looking at a single time point. And stuff is cleared up so rapidly by these cells that you often don't even notice it. Having introduced you to their fantastic phagocytic capacity, let me move on to talking about TREM2, which is a phagocytic receptor. And here it is on the accumulating around the phagocytic cup of a macrophage that is engulfing a dead neuron. So all of this work has been done by Hazel Hall Roberts in our lab, who is postdoc between our lab and the Alzheimer's Research UK Oxford Drug Discovery Institute. This work is now in press, so hopefully in a few weeks' time you'll be able to read it for real. So TREM2 is has the second highest odds ratio for Alzheimer's risk after ApoE4, and R47H is the most common mutation in TREM2 in Alzheimer's, although there are others. And TREM2, as I've said, is a microglial phagocytosis receptor, and its downstream signaling pathways are still being precisely worked out, but generally we know that it's involved in pro-survival signaling and anti-inflammatory signaling. And the R47H mutation is in the extracellular domain in the basic sort of cup, which is involved in binding to anionic ligands such as phosphatidylserine and ApoE and A-beta oligomers. It can also be involved in, as well as phagocytosis and survival, inflammation, questions over chemotaxis as well. All of these phenotypes that have been assigned to it up till very recently have mostly been as a result of the mouse, modelling it in mouse, where I've already said that it was found by Christian Huss's group a couple of years ago that the R47H mutation leads to a splicing defect in mouse, but not in man. And so that means that in mouse, it's effectively a null mutation. And so we have to sort of revisit a lot of the mouse data and flesh it out with human data, hence the relevance of using human iPS cell microglia to model this. There's also been a humanized mouse model developed with human TREM2 on an AD background. And that has shown that if you have knockout of human TREM2 in that model, then there is reduced microglial clustering around amyloid plaques. But the R47H mutation doesn't follow the same pattern as the knockout. It's more like the wild type. Similarly with human ES microglia, it's been shown that the TREM2 knockout leads to reduced ex vivo plaque clearance by microglia, but the R47H mutation does not follow the same pattern as the knockout. It's more like the wild type again. So those two studies seem to sort of confirm each other. And that's it as far as iPS microglia studies of TREM2 are concerned, apart from another really interesting study done by the Pocock lab showing that R47H has, and other mutations in TREM2 lead to a locked metabolic switch, which this paper came out very recently, and it's by which they mean that microglia and macrophages, under normal circumstances, they are using oxidative phosphorylation. But when they're activated, then they switch to glycolytic metabolism to be able to produce at very short notice enough energy for all of the inflammatory functions that they need to undertake. And that this switch seems to be locked in TREM2 R47H mutation. So that's really interesting. That's something rather different from what we've been looking at. So Hazel, we are using an isogenic series of iPS cell lines which have either wild type TREM2 or R47H knocked into both alleles, or biallelic knockout. So using that isogenic series, Hazel has explored all of these different functions of TREM2. So surface expression of TREM2, and then the downstream signaling pathway from TREM2, and then all of these phagocytic and other functional outcomes. And also looked at RNA by RNA-seq at the transcriptomic changes in the cells. So first of all, to look at the surface of TREM2. So TREM2 is expressed on the surface, but it can be cleaved by proteases such as ADAM10 or 17, and this leads to release of soluble TREM2, which is, we don't really understand what its function might be, but at the very least it means that there's less full-length TREM2 on the surface of the cell able to undertake normal signalling. We found in our IPS model that R47H expressing cells had less surface TREM2 on them than the wild type cells. And this was by a very sensitive biotinylation and pull down assay, which is rather more sensitive than just looking by FACS or immunocytochemistry. Also by ELISA found that more TREM2 was shed from the R47H cells than from wild type cells. So these two sort of affirm each other's. And it's slightly surprising because it's not been particularly reported for this mutation that there should be a difference in shedding, but this is what we have seen in our model. Then she looked to see whether the signaling pathway was intact in these cells, and indeed it is. So if you activate the TREM2 with an activating antibody, then it leads to phosphorylation of SYK, and this is shown here. This is an isotype control antibody that will not activate TREM2. And then further downstream, we can look at the calcium response of the cells, and this too is intact in our cells. So the whole TREM2 pathway is intact in these cells, which makes it a relevant model. So then we started looking at downstream functional readouts. And the most obvious one is to look at phagocytosis in the cells. And so using a nice assay that she developed, looking at the uptake of Frodo-labeled dead neurons, could see the association of TREM2 with the phagocytic cups when the cells are being phagocytosed, but that there is no difference between the R47 mutation and wild type uptake of these dead cells, although the knockout cells do have reduction in the amount of neurons that they were taking up. So in this functional readout, R47 is close to wild type rather than to knockout. She did a whole battery of different functional phenotypes, and it's basically the same kind of story as for phagocytosis, so also survival without MCSF and C5a mediated chemotaxis. These TREM2 knockout showed a decrease, but R47H mutation didn't phenocopy the knockout. It was pretty much like wild type. So no gross phenotypic differences with the R47H mutation then in our cells. So then we moved on to looking at transcriptomics, and for this experiment took the 3 different genotypes and took three replicas of the differentiated cells, and by the principal component analysis, you can see that actually the first principal component that we found on analysing this data was the differentiation age of the cells. So this is quite an interesting cautionary tale, is that these differentiation cultures, they're highly productive and they keep on spitting out macrophage microglia precursors for many, many weeks. We've even kept them for up to a year. And although we do our experiments within a very short, small window of 7 to 9 weeks, each week the cells are clearly changing in their transcriptome, enough for us to be able to detect it very obviously on these types of transcriptomic experiments. Luckily, the bioinformaticians are able to correct for this. So in the downstream analysis, it's no longer an issue. But yeah, it's something to take home from this. Second principal component on this axis is the genotype, as we would expect. So looking globally at the differentially expressed genes, we see that the TREM2 knockout has a lot of differentially expressed genes versus the wild type cells, both up and down. The TREM2 R47H mutation cells has a small number of genes that are differentially expressed either up or down. And importantly, the majority of those differentially expressed genes from R47H are also shared in the same direction by the knockout cells. So this is a good demonstration that R47H is following the same transcriptomic pattern as the knockout, but just to a lesser degree. We were able to dig out some leads from the transcriptomics to follow up, and integrins seem to be a group of proteins that were particularly down-regulated in the R47H and TREM2 knockout cells. And so here's a validation by qPCR of integrin beta 3, which is down-regulated in the knockout and also the R47H. This was also further validated by Hazel by flow cytometry, so on the actual protein level, and we see the same pattern. Now, interestingly, TGF-beta is also down-regulated in the knockout cells compared with wild type, and this must be a TREM2 dependent phenomenon, because when you add a Syk inhibitor, which is just downstream of TREM2, then that reduces the amount of TGF-beta that's produced by the cells. So we reasoned that, well, maybe if you increase the amount of TGF-beta in the culture, maybe it'll rescue these expression of adhesion proteins. And indeed it does so. And so this integrin alpha V beta 3 is upregulated at both the transcriptome and the protein level when it's when the cells are incubated with TGF-beta. So then Hazel went on to look at how this translated to cellular adhesion, because alpha V beta 3 integrin binds to vitronectin preferentially, and so she developed an adhesion assay for this, and indeed the knockout and the R47H cells have reduced amount of adhesion to vitronectin, and this is further validated by using an inhibitor of the alpha V beta 3. Now it was then the next obvious question was, well, does TGF-beta rescue this? Unfortunately not, which is slightly disappointing, but that's the way of things. So clearly TGF-beta, although it's able to rescue the protein levels of the beta 3 integrin, it's not able to rescue adhesion, presumably because there's many other things that TGF-beta is doing as well in the cells at the same time, and so it's not a very clear-cut phenomenon. But at least it does give us a lead that integrins are important and dysregulated in these Alzheimer's associated mutant TREM2 proteins. So to summarize this section, R47H is shed more readily than wild type in our in vitro iPS cell model. The R47H cells do not display these gross defects in phagocytosis or chemotaxis or survival. Instead, it has a much milder transcriptional dysregulation to the knockout. So this supports the hypothesized partial loss of function. And it shows us that transcriptomics can guide us to these more subtle R47H phenotypic effects, and such as cell adhesion to vitronectin, and it can help us to predict targets for therapeutic intervention. So this leads me on to the final part of the talk, which is about LRRK2, and we know that LRRK2 is downstream in it's not a phagocytic receptor, but it's involved in downstream events in the phagocytic pathway, which I'll show you more detail of. So it's implicated in immune function. It's a large and complicated protein with many domains, a kinase domain and a RAS-like GTPase domain, and various protein-protein interaction domains. It's expressed in many immune cells, upregulated with gamma interferon, and it's associated not only with Parkinson's disease, but also with mycobacterial infection and inflammatory disease of the bowel. Its protein structure has been very recently, over the summer, shown by cryo-electron tomography, to show that it can bind to microtubules in a rather beautiful fashion, but it doesn't always bind to microtubules. Whether it is actually expressed in microglia has been a little contentious. There's not much evidence, but there is a bit of evidence. For example, this paper shows LRRK2 positive reactive microglia in dementia with Lewy bodies. It may be that LRRK2 is only expressed in these microglia when in a pathological condition, when there's been upregulation of the gene. It's certainly expressed in our iPS microglia, which we can see here, more so than in the neurons that it's embedded with. So the LRRK2 work that I'm going to talk to you about has been done by Cecilia Lee, and who was a PhD student in our lab, and this has been recently published just a couple of months ago. So the basic protocol for investigating LRRK2 localization in the iPS macrophages and microglia was to take the iPS-derived cells, trigger them with interferon gamma to increase expression of LRRK2, and then challenge them with a phagocytic meal, and then look by high content imaging at what's going on within the cell. It was immediately apparent when we fed them yeast zymosan particles that LRRK2, which is normally just sort of distributed around in the cytoplasm of the cell as these little red dots, accumulated on some of these phagosomes to varying degrees, and sometimes not at all. This specificity of the stain was for certain, because in the knockout cells, knockout LRRK2 cells that we had made, no staining at all. So what is special about the particular subset of phagosomes that LRRK2 is localizing to? Well, you can track the maturation of phagosomes by the recruitment of various different markers to the phagosomes as they mature. And so we could see that early phagosomes that are labeled with RAB5 were not associated with LRRK2, but the later maturing phagosomes that are labeled with RAB9 and or LAMP1 are labeled with LRRK2 as well. So this shows that, and this is the quantification of it, that LRRK2 is localizing to maturing phagolysosomes. So at the point around the time when phagosomes are fusing with lysosomes. So we wanted to know more about what's going on at this time point and what the interacting partners might be of LRRK2. Around the time that we were doing this, Dario Alessi published beautiful papers showing that LRRK2 can phosphorylate a subset of RABs, quite a long subset of RABs. I mean, it's a big class of proteins. There's at least over 40 members, and a subset of about 7 or 8 of them could be phosphorylated by LRRK2. And RABs are responsible for shuttling vesicles around in the cell. So we wanted to know which other RABs might be involved at this point in the maturation pathway and what the downstream consequences of that might be. There aren't good antibodies for all of the RABs, and we're specifically looking at endogenous protein, not exogenously expressed protein. So we're rather reliant on antibodies. But we were able to show that RAB7 is not co-localizing with LRRK2 coated phagosomes. Here's a LRRK2 coated phagosome that looks like it's got a balaclava on, and RAB7 is clearly not recruited to that. However, RAB8 is very strongly recruited to LRRK2 positive phagosomes. And similarly, RAB10, although this antibody is not quite so good as the RAB8 one, is also recruited to LRRK2 positive phagosomes. And here is the quantification of it. So then we asked, well, is it necessary for RAB8A and RAB10 accumulation on the phagosomes? And so by using a knockout of LRRK2 line, if you have no LRRK2, then you also have no RAB8 and no RAB10 being recruited to the phagosomes. And there's the quantification of that. So then we wanted to know whether kinase activity of LRRK2 is responsible or required for RAB accumulation on the phagosomes. And there are very good LRRK2 kinase inhibitors. We used a couple, GSK and GNE. And the first thing that we noticed was that when you applied these inhibitors, LRRK2 is actually, it even increases on the phagosomes. So we get what we called supercoated phagosomes. And here is the quantification of it. So the number of...
note that we've only got like 2 minutes left.
2 minutes left, yes. So LRRK2 is still recruited to the phagosomes at the same rate, but they become supercoated. But despite this supercoating, we get less or a massive reduction in the amount of RAB8 or RAB10 that is recruited to the phagosomes. So it is dependent on LRRK2 kinase activity. And further, there's a fantastic antibody against phospho T73 RAB10, which confirms this, that the RAB10 is phosphorylated at the phagosome. And there's a beautiful balaclava there of phospho RAB10 on the phagosome, and there's the quantification of that. So to summarize all of this, well, luckily it's been done by others who have worked on very specific parts of the pathway as well. LRRK2 is clearly, it's phosphorylating this subset of RABs at the phagosome membrane. And when it does so in a way, in a position on the switch 2 loop that changes the RABs from being able to interact with one set of effectors to interacting with a different set of effectors instead. And these downstream effectors is something that we'd like to investigate further next, because we don't know what's happening. It sometimes looks like vesicles may be being streamed off from the phagolysosome, and we're curious to know where they're going and what they're doing. They could even be recycling stuff back out to the surface, which, if it was alpha-synuclein or tau, would be potentially very interesting. We're not the only ones who are looking at this, of course, and people have looked not in the same way at phagolysosomes specifically, but they've looked at lysosomal stress, which can be induced by things like membrane damage and leads to calcium leakage, and found a whole bunch of different downstream effectors of RAB8 and RAB10. So all of these pathways may be implicated in our model. So our overall summary is that, well, I hope I've shown you that our cells are, our IPS model can be used to look at the function of the gene products of genes that are involved in neurodegenerative disease. I specifically shown you some evidence around TREM2 and the role of, and the phenotype of R47H TREM2. And then further downstream in the phagocytic pathway, I've shown you how LRRK2 is recruited to phagolysosomes and recruits RABs. And I hope that this model will be of use for integrating a whole load of these other disease implicated genes into these pathways that are so important to microglial function. So I'll leave it there and take some questions. Thank you very much. I'd like to just, I guess, finally thank everybody who's been involved in this work. This is a socially distanced picnic that we were able to have in the summer before lockdown has come back upon us. I'd like also to thank all of the donors of the IPS cells in the first place, and all of our funders as well. So thank you very much for listening.
Great, thank you. So I have just got to unmute Guy, and then he will ask you some questions from everyone.
Great, thanks.